Guess it got quiet enough. We could start. um, Thank you all so much for joining us here. This is the last 2024 seminar of the Biotech Connector speaker series. We try to have these quarterly. This is done in connection and based on a partnership we have with the Frederick Chamber of Commerce. So we're very excited to have them support ongoing support for the Biotech Connector. I am Maggie Scully, Director of Partnership Development Office here at the Frederick National Lab for Cancer Research. Our aim is to enable connections with the community, enable connections across disciplines and scientists, and also connect with the Frederick National Lab investigators. So a little bit more about this event. It's a quarterly series to promote Frederick County and the surrounding areas, biotech and bioscience community. The aim is to allow for networking, which is great to see already happening. It also allows us to get an inside look at some of the advances and really welcome you here into the building. And again, bring this opportunity to network across scientists and entrepreneurs and our community and biotech-minded folks. I'll have a brief slide. Frederick National Lab is a collaborative enterprise. We're interested in partnering opportunities. These are contact information to the partnering office. We also connect with external organizations through subcontracting, and their website is there as well. And there's more information on the frederick.cancer.gov site. And also some of you may be here to learn about different careers, and we have a number of different career opportunities here at the Frederick National Lab, and that is the website as well. You're welcome to please visit. If you have any questions, please reach out to myself. There's a number of other FNL folks that can help you as well. So now we get to our three amazing speakers. Our topic today is next generation sequencing technology. Our first speaker for today's event will be Mr. Bao Tran. He's the Director of Operations at the National Cancer Institute Sequencing Facility. He supervises a team of bioinformatics, quality control, research associates, and R&D scientists, and is responsible for the day-to-day operations of the sequencing facility. He serves as a liaison between the National Cancer Institute labs and the sequencing facility to develop strategies and complete projects with high-quality results. He obtained his master's degree in biotechnology from Johns Hopkins University and a bachelor's from George Mason University. He came to FNL, to the Frederick National Lab, from the Craig Venter Institute, where he was a scientist in the research and development group for over 10 years. At the Craig Venter Institute, he focused on developing new methods to optimize library constructs for Sanger sequencing and next-generation sequencing. So we're very happy to have you here as our first speaker. The second speaker will be Dr. Olena Lar. She earned her Ph.D. in molecular genetics from the Institute of Molecular Biology and Genetics, the leading molecular biology research institution in Ukraine. After moving to the US as a postdoc fellow, Dr. Lar worked in both academic and industrial settings. She ran a biotech startup and eventually transitioned into the diagnostic space due to the COVID-19 pandemic. In 2021, she led the establishment of the Next Generation Sequencing Department at CIAN Diagnostics. Her team has integrated both short and long read sequencing as well as traditional Sanger sequencing into their offerings, achieving flexibility, cost efficiency, and customization to meet the needs, specific needs for clinical and research applications. I'll move this here just so you have it. And our third speaker is Dr. Samuel Rulli. He's an Associate Director of Global Product Management for Next Generation Sequencing and Applications at QIAGEN. Dr. Rulli received his PhD from Tulane University studying the gastric proton pump and did postdoctoral research at Johns Hopkins University and NCI Frederick. Down the road from us. Trained as a molecular biologist, Dr. Rulli has worked on different assay detection technologies for gene expression and nucleic acid analysis. So I hope everyone enjoys the conference. If you have any questions, please feel free to reach out at the end. We should have time for two or three questions after each presenter. And for the folks online, you can submit your questions in the chat box or unmute yourself and we'll have someone, we could either have someone ask the question or ask you to unmute. Again, for those folks online, please stay muted during the presentations. Okay, now we're here to please welcome our first speaker, Mr. Bao Tran. Thank you. Okay, hold up. Yes.
It is a pointer? Good. All right, good morning, everyone. My name is Bao Tran, Director for the Sequencing Facility, and thank you so much for having me today. So today I'm going to break my talk into two parts. One is the quick introductions of my facility operations, and then we're going to dive in a little bit to the surface of the next-gen sequencing center technology and application. All right. With that, really quick introductions of our facilities. We are a high throughput sequencing facility for the NCI. And we do have access to, obviously, it's the sequencing facility, so we do have access to quite a number of, going back, to the number of high-tech sequencing or state-of-the-art sequencer currently on the market, such as this is very sensitive. The NovaSeq 6000s, NovaSeq X Plus, the applications that range from DNA applications to DNA RNA applications, transcripts, profilings, gene expressions, as well as whole genome sequencing target. Again, very sensitive. Don't know why. But anyway, so those are for the short read technologies. For the long read, we do have the PacBio operations in our lab. For long read is the PacBio and Oxford Nanopore offerings long read technologies for applications such as the novel assembly, target sequencing, base modifications, and structure variation using the long read. Besides sequencing technology, we do have the optical mapping technology in our groups to use for a very large structure variations. We are talking about megabase, hundreds of kilobase to megabase structure variation, whether it's deletion or insertion, copy number variation as well. We do offer single cell capability in our lab as well, using both the 10x genomics as well as Mission Bio for target DNA sequencing, single cell, sorry. So as I mentioned before, our lab is a very high throughput, relatively high throughput facilities. We serve roughly about 260 NCI PI within last fiscal year, processed roughly on average about 14,000 samples in the last years or so. And the applications is ranging, like I said, most of the applications are dealing with the, sorry, I cannot use the pointer because it seemed like it advanced in the, all right, good. So most of the applications are for short read technology, so that including the RNA-6, mRNA-6 for coding RNA, whole genomes, sequencing, and exome. This is a really interesting point here. It used to be the exomes, we sequence more exomes than whole genome, but because the cost of sequencing is going down, thank you so much, the cost of sequencing has went down quite a lot in the last few years or so, especially with the high throughput sequencer that we have. We're seeing more and more people utilize the whole genome sequencing instead of exome, which is good because you got a lot more information out of whole genome instead of exomes. So this is the point that I try to make here. So this is the number of gigabase or billion base pair that we deliver every year. As you can see from FY23 to FY24, our throughput went up almost double. That even though our number of samples are not increasing that much, that is due to the newer next generations sequencer that we have, higher throughput, the NovaSeq X Plus, the PacBio Revio, as well as the ONT P24 that we have in the labs. All right, so one of the things that we, as a high throughput facilities, we put in a lot of emphasis into ensure that our quality is, as you probably know, even though the cost of sequencing is going down, but the cost of the cost of sequencing is still relatively expensive operation. So we want to ensure that the PIs has their most highest quality that they can have for the money that they pay. So with that, we put in a lot of qualities control throughout the entire process. From the time that the sample are submitted to us, we immediately run the quality control ensure that no garbage in, no garbage out. After the library construction, we do another quality control to ensure that the quality of the library is good enough to go into sequencing. And we run qPCR for quantification in terms that we need to pull samples together, as well as every single run that we run, whether or not it's short read or long read, we always have control sample that's spiking into the sample itself, whether it is 5x or lambda into our sequencer to ensure that everything that we sequence will be matching correctly or high quality for the run that we sequence. And obviously, our informatics team developing quite a comprehensive data quality check after we finish sequence before we deliver the data to the PI itself. So as you can see the number right here, from last fiscal year, if any sample that passed quality control, 99.3% of those samples will be successfully sequenced and deliver high quality. Beside those, since we are a high throughput facility, we do have a very comprehensive LIMS system in our labs, our lab information management system, for those who are not familiar with the terms. And it keeps track of all the samples that within our facilities. As soon as the samples are coming into our labs, we are immediately tagging it or generate a barcode, and that sample will be able to track throughout the entire process in our lab, from the sample submission all the way to, I don't have it here, but all the way to data delivery. So like I said, we are really keeping track of all the project and all the samples that come into the lab, and it ensures us to not only keep the sample within the pipeline, but also ensure that we can be able to know where the resources are needed to move people around or moving resources around. Such as you can see here, if you've seen that the RNAs have a lot of sample coming in, but we don't have any sample for TruSeq, then we can move people around or move resources around if needed. All right, so everything we've done here means nothing if we cannot deliver data. So we do have informatics team that embedding with us, and we developed a very comprehensive data delivery package that including the standard QC report for all the samples that hit in our lab using the multi-QC. On top of that, we generate a very comprehensive data delivery package that including all the information related to your sample or the project itself. Our goal is for the PI to be able to take this information and be able to just plug and place into their paper. That way, they can be able to develop in the method session without having to go back and rewriting anything or rewriting anything that they need. That including all the the sample information, the pipeline for the informatics that using to analyze the project itself, as well as the summary of all the data. We don't do in-depth analysis because we don't have enough bandwidth to do that. But at least for secondary analysis, we providing very much information needed for them to just plug and play into their pipeline. So in terms of deliverable, obviously we deliver all the raw data file, multi-QC report, MATPEM file, gene quantification, if it's RNA or DNA, single cell, we provide in Cell Ranger, as well as variation or SV or CNV depend on application. And obviously for ATAC6 or CHIP6, we deliver the peak calling as well. Assemble and annotation is depending on project. Most of the times is for R&D project or R&D purpose. All right, with that, let's dive in. How I do in time? Good? So gonna be really quick in terms of next generations and what we have in the labs. So the next generations, as you probably know, is sometimes called short read technology. And this is the current technology that we are using. So I don't know why they still call it next generation, which is kind of funny. But anyway, so the reason for that is because the short read technologies are sequence billion of base pair, million to billion of base pair. Short read. It really short is a few hundred base pair, but we sequence massive amount of it. So the advantage of short read is obviously because we can be able to sequence so many of them, the cost is really, really going down. And it's extremely fast. We can be able to complete a human genome in a few days. And it's a very high throughput. So high throughput means high sensitive. So you have more confidence on the read that you are calling. And obviously, because the technologies is really developing in the last 10 years or so, there is a really large number of applications that are out there, anywhere from molecular biology all the way to forensics, using for forensic science, as well as virology. We just went through the pandemic, so you probably know how important the next-gen sequencing was placed in the pandemic itself. However, short read does have some disadvantage. One of the obvious disadvantage is a short read. It's really short. It's a few hundred base pair. So because of that, you really need to have a reference genome to ensure that you can be able to map the read back. You can use it for smaller genome without a reference genome, but anything larger than a million genomes and things like that, then you really need to have a reference genome for that. Because of amplification, all short read technologies still need to have an amplification step. So it relatively poor sequence for an area of stretch of DNA that have repeat single nucleotide that repeating homopolymer. And like I said earlier, all short read technologies are still rely on the PCR amplification, whether it is bridge amplification for the Illumina or rolling circle amplifications for all the technologies such as the MGI. And because of the PCR amplification, it does have problems with PCR bias and GC contents, and obviously PCR does have base error that you have to account for. All right, the current short read technologies are monopolized by a company called Illumina. If you are anywhere near the sequencing technology currently or doing any sequencing, change are you gonna using their technology, the Illumina technology, or sequencer to do that. So they are currently, like I said, almost to the point of nearly monopolize the sequencing market right now. The reason for that is because, first of all, because their instruments are relatively easy to operate, as well as they have a very simple, relatively simple, relatively simple, the pipeline for library constructions as well as the sequencing itself. And obviously, since they are leader in the sequencing market right now, they developing quite a large number of applications that you can use their sequencer for, that including whole genome, whole transcriptomes, all the way to ChIP6 single cell. You can do it for target capture, ChIP6, ATAC6, and they have many, many more. As the list of the application that I show you, we have more than 24 applications currently in our lab that's supporting under the Illumina pipeline. All right, in terms of the platform that we have, Illumina platform that we have available in our labs, we have a large range of the sequencer that going from anywhere from the I6 that only generate a few million base pair, all the way to the really high throughput, the NovaSeq X Plus that we just implement the last 2 years or so, years and a half, and that generate almost 50 billion read in terms of output. So it's so that why the reason that we have so many of this in our lab is because it application. So if you only have a very small set of sample, you don't really need to have a lot of read, then there's no reason for you to use the high throughput machine because it costs a lot to running on this. It still costs about $16,000 per run for each of the flow cell here that we are running. However, if any of you remember the first human genomes that sequenced using the Sanger, it cost about $1 billion and about 10 years to complete the first genome. With this instrument right here, we can do 128 human genome in 48 hours, and it costs $250. So the cost of sequencing has been going down quite a lot, but still relatively expensive in terms of if you have a lot of sample or a lot of project to go through. Okay, so those are the short read technology that we currently have in the labs. Let's move on to long read. So we have no imagination in our labs, so we call short read and long read. Why? Because it's just longer. So long read is sequence in terms of short read is only a few hundred base pair. For long read technology, we actually can sequence from KB to from a few thousand base pair to megabase, million base pair, depend on instrument or application that you need. So one of the reasons why short read is really good at resequence something. But if you want to stack sequence, oh, you know what? Let's do this. So, so why is long read is important? So with short read technology, what you do is very much like this. You take a genome. In this case, Christmas is coming, so we use in Christmas tree. You put it through a chipper, and then you take all these pieces that generate out, and you assemble them. You put them together, and this is the output. You're lucky to have a tree or something similar to a tree. If you are that lucky. Normally you don't. If you don't have a reference genome, you end up with something totally different from what you start with. All right, so why long read? So the same thing. You take a Christmas tree. This time, instead of using the chipper, you're gonna have Captain America and Iron Man to help you chopping the tree down. So it chopped into a much larger piece. With this, you're gonna be able to get much closer to your original target. In this case, it's the same family, so that's good. So short read and long read, that's very much the difference between short read and long read. The other advantage of long read technology is that it sequences a single molecule, which means that there is no amplification step in there. Whatever DNA or RNA you give us is what we're gonna be sequenced. We don't do any amplification. So with that, it's gonna help with the GC bias or TC bias. You no longer have to worry about amplification errors and things like that. And because it sequences native DNA or native RNA, you can be able to detect modification directly without having to do any of the bisulfite conversion to convert your DNA into a C to T, right? In order to detect modification. However, there's no free lunch. The long read technology does have some disadvantages. One of the biggest disadvantages is that the long read does have higher error rate compared to the short read technology, but they have way to overcome that, and I will discuss in a second or so. And obviously, it generates much less data output compared to the Illumina, but that is not so bad. The new technology, the current instrument that we have in the lab actually generate quite a lot more. It's not to the point that it's going to be compete against short read, but it gets in there. So because of that, the cost per base is relatively higher than the Illumina itself. And obviously, it does have a longer runtime compared to the short read technology. The current two technology for long read in the market currently, and we have both of them in our labs, is the PacBio. PacBio is the probably the first commercial long read technology out there. There's another company called Helix, something like that, but they die out. So PacBio taken over. Currently, we can be able to generate roughly about 20 kb high 5 read or high fidelity read for the PacBio compare. So 20 kb means 20,000 base pair compared to a few hundred base pair for the short read technology. So it's still relatively long. And again, there is no amplification step. The most important I'm highlighting here is all the error that generate from the single molecule sequencing is a random error. The reason why it's important is because the way that the PacBio read in the molecule or generate the library is a circular molecule. So when the DNA are deposited into the ZMW or the sequencer, the sequence module, the polymerase are anchored at this is a live polymerase. It anchored on the bottom of the ZMW, and it will sequence the DNA. And it will sequence the DNA over and over because it's a circular molecule. So it keeps sequencing the DNA over and over and over and over. So when you finish with that, you can stack them up and do the consensus sequence read. So because the error is random, so you no longer have to worry about the same error that making by the amplification step that causing the same error. So which means that you can be able, now you can call consensus sequence and it's very accurate. It's as accurate as the Illumina right now in terms of the read for the PacBio itself. But because you have to read the molecule over and over, you no longer can assist 20 kb. 20 kb is kind of the max that you can be able to do right now for PacBio. So the key advantage is with a much longer read, you can be able to do de novo assembly for whole genome. You can do really long target sequencing that using for long amplicon, HLA, full-length HIV, or full-length vector sequence. You no longer have to do assemble anymore. And the most important, or the biggest application that we are using the PacBio for right now is the transcripts isoforms, which means that now we can be able to sequence the isoform from start to finish without having to break it down into smaller pieces. Because of that, you can be able to identify isoforms from the transcript itself. It's a very powerful application that we currently offer in the labs. All right, and another application is another long read technology is Oxford Nanopore, so ONT. So it's a little bit different from any other technology out there. The reason for that is that it does not use any camera or laser or anything like that to capture the base pair. It's actually just pushing the DNA through. It's like science fiction, pushing the DNA through a nanopore, and they measure the current difference from when the DNA or the molecule or DNA or RNA going through the pore, and they measure the differences in the current, and that will be registered. And you sequence long enough or you have enough machine learning, then you can be able to stack them together and identify the base sequence for that. So again, the ONT can sequence a really long molecule, much longer than the PacBio. We can be able to sequence one read, but we still count that, 2 megabase, 2 million base pair. So it's really amazing that they can do that long. And again, no amplifications, no chemical labels, anything like that. Because of that, we can be able, again, just like PacBio, we can be able to detect modification for DNA. However, ONT is the only technology currently that can be able to sequence RNA directly. So you don't have to convert RNA into cDNA. You just take the RNA and sequence them directly. Because of that, we can be able to detect RNA modification as well. It's a really cool application. So anyway, so this is what I'm referring to. Most applications are very similar to PacBio. However, with ONT, you can be able to detect the direct RNA-seq, and detect M6A base modification for that. It's really good applications for target sequencing as well. The reason for that is because you can use Cas9 and be able to cut the RNA and sequence directly on that. I know that I'm running out of time, so I'm not gonna go through the other two applications that we have in the labs. But if I'm staying behind, if anyone want to talk to me, I should be behind. That is the single cell. I'm just gonna roughly saying that we do have a single cell in our lab, that including the 10x Genomics, Mission Bio for DNA, single molecule DNA, and we start working with the PIP6 as well. This technology is amazing because it's cheaper than the 10x as well as instrument free. So any lab can do single cell now. Long optical mapping technologies. This is a really cool technology for those who are looking for extremely long CV, SV, and CNV. This is application that you can use for looking at the whole genome SV. And unfortunately, it's not a sequencing technology, which means that you don't have base pair resolution, but at least it gives you a really nice picture of where your modification is, your SV is, and it can go much larger than the sequencing technology currently have. So at least you can be able to know where it is, and then you can zoom in and using the long read technology such as PacBio to fill in the gaps, basically. So one of the things that people have been asking us all the time, so what is best? What is the best out there for use? It's all dependent on your application. It's not really depending on the sequencer itself. It depends on your application. What do you want to do? What do you, the end goal for you? With that, we can be able to show you or at least guide you to what the best technology to use. So again, if you want to learn more, to talk to us, we probably know a thing or two about sequencing. All right, this is my team. Thank you so much for your talks. For your patience. And this is my team. They actually the one who doing all the work. I'm just here for show and tell. All right. Thank you, guys. Question? Do we still have time for question?
Everyone? Who are
Yes.
your clients?
I'm sorry?
Who are your clients?
Oh, my client is NCI, so NIH. We can offer service, only can offer service within the NIH currently, but we can always collaborate in terms of helping. If you need anything, we can always guide you to it. We might not be able to take your project in, but we always can work with you to help you with whatever you need, answer any question you have. And we've done that a lot, actually.
Thank you.
All right. Yes.
Is there a sequencing method for AAV?
I'm sorry?
AAV genomes? Gene therapy?
I'm not sure, but why don't we chat after?
I have a question.
Yes.
Regarding the homopolymer.
Yes.
Repeating difficulties with a short read.
Yes.
How do you get around those challenges from a bioinformatic point of view? Because I'll see if it's like a stretch of 4 to 8, I have anywhere between maybe 3 to 10 might be there.
Yeah.
At the end of the day, most of my whole plasmid sequencing, I do get expression.
Yes.
Right. So it is a functioning plasmid, but how do you deal with that bioinformatically?
We use long read.
Ah. Okay.
Oh, neither that or our. I don't have our informatic teams here. Actually, I do. So they probably have tools that can help with that. But most of the time, if you have a very long stress of repeat or homopolymer, we just throw it in our long read.
Okay, and then your long read, because you have so much coverage over it.
Yes.
You know that it's
you have million of coverage over that, so you can roughly overkill it, but still, it's easier than spending time trying to figure out which one is which.
Excellent. Thank you.
Yeah. Okay. Thank you so much, guys.
So,
Yeah, just switch to the next
Sure.
slide.
oh, okay. So yeah, good morning. My name is Olena Lar, and I'm Senior Director for Research and Development
as well as a founder and the head of the next generation sequencing department for CIAN Diagnostics. I'm going to talk today a little bit about innovative approaches that we develop at CIAN Diagnostics to make genetic testing more accessible and cost-effective. So first, a few words about CIAN Diagnostics. It started in 2013 as a toxicology and general clinical chemistry lab, and at the time of pandemic, majority of our efforts went into COVID-19 testing, for which we've got a number of recognitions from the state of Maryland. But then with the COVID-19 samples diminishing, we expanded our offering, and currently we are full spectrum diagnostic lab offering everything that's your typical diagnostic lab does, including genetic testing. So let's take a look at our next generation sequencing department first. It started in 2020 as a response to the emerging need for sequencing, whole genome sequencing for COVID-19 virus to detect new and emerging variants. And we started this department from scratch with just one MySeq instrument, and then quickly validated COVID-19 whole genome sequencing for variant detection using Illumina technology, and then cross-validated for the use with Oxford Nanopore technology, achieving 100% agreement between two platforms. So currently our department consists of a number of Illumina instruments, as well as Oxford Nanopore instruments and ABI genetic analyzer for traditional Sanger sequencing. We've developed a number of genetic tests, which we are currently offering for our clients, and we're also working on some innovative solutions for genetic testing. But first, let's just go briefly about what genetic testing and why we are doing this. Basically, genetic testing is evaluating a person's DNA to identify variations that lead disease predisposition, differences in traits, and it also currently becoming very important direction in directing targeted treatment choices based on your genetic landscape. So it's worth noting that if you look at human genome, 99.4% of human genome is stable. It's what shared among all the humans, and only 0.4% of human genome is variable. And this is what we are evaluating for genetic testing. Typical majority of these variants, they consist of single nucleotide variants. It's just a one-letter replacement in your DNA sequence, insertions, deletions, as well as a large structural variations that are a big rearrangements, typically chromosomal level. So when we are doing genetic testing, we're using next generation sequencing technology. And what made next generation sequencing so widely acceptable and making a breakthrough in genetic testing? It's basically three things. It's throughput, speed, and scale that we can sequence now. Basically, next generation sequencing is a massive parallel sequencing, allowing for analysis of millions of DNA fragments simultaneously, allowing for a large scale project, as well as using next generation sequencing in any applications from clinical to research. As Bao just pointed out, when we think of next generation sequencing, we typically think of Illumina. Illumina made genetic testing while the world was accepted based on that it's offering standardization in platforms, library preparations, and offering very robust and user-friendly bioinformatic tools, as well as it's earned its recognition as a golden standard in next-generation sequencing for its high accuracy, achieving 99.9% accuracy. But it has certain drawbacks in terms of being used in clinical diagnostics. First of all, Illumina instruments have a very high capital cost, and they are typically large, so they rely on developed infrastructure. Also, it makes using it for smaller lab and remote location really difficult. On the other hand, if we look at Oxford Nanopore technology, again, very good described by Bao in previous talk. I just want to show you the scale of it. This is Oxford Nanopore instrument. Basically, it has almost zero capital cost. When you purchase it, you pay only for the cost of the reagents. You get the instruments for free. So it's very easy for even a smaller lab to start doing next generation sequencing. It also you can take it to the field basically anywhere. It's been taken to the space station for the analysis. It does not require any infrastructure. Also, it's very cost effective in terms of cost per sample. So it can be a game changer for a smaller lab, for rural area, for remote areas, or for the field. Also in events of pandemics, it's very easy to just start massive sequencing anywhere, basically. So in our lab, we are always thinking of how to make genetic testing more affordable, more widely available, and so we can reach more people with this. One of the projects that we have in our lab, we are looking at comparing the performance of Oxford Nanopore versus Illumina for a particular type of genetic testing. So for the pilot study that we've done, we did a pilot study using 3 human samples, for which we used validated in our lab cardiac arrhythmia panel. It's amplicon based. So the experimental design was that we took this, each of the samples, and we took it through in parallel through Illumina library prep and Oxford Nanopore library prep. We ran it on a corresponding sequencer, did the specific for the platform analysis, and then we were comparing variant call files to see the concordance and discordance between the platforms. Once we analyzed that, we randomly selected several of the discordant variants to do a Sanger confirmatory sequencing. So this was the outcome of this small pilot study. We've got between 89 and 90% agreement between platform. Majority of variants agree, but then each platform generated some number of unique for that platform variants. So this is where it became really interesting, and we took a closer look at those unique variants for each platform. So first, I have a few examples here for you. One of them was a unique for Illumina variant, where Illumina calls for, how did this one work?
On the top.
On the side. I'm sorry. Oh, I see.
Mhm.
No? No?
Oh, yeah.
Oh, cool. Oh, okay. So Illumina, in this case, Illumina calls alternative C variant here, where Oxford Nanopore calls reference G. So we use this for Sanger confirmatory testing. And in this particular case, Sanger sequencing supports ONT call. So from both forward and reverse strand, we get the same call. So the next one was a very interesting case where it's a repeat region and all sequencing platforms are not great at resolving repeat regions. So in this interesting case, we have Illumina unique variant next to Oxford Nanopore unique variant. So Illumina calls for alternative G here, whereas Oxford Nanopore says it's reference A. But on the same at the same time, ONT calls for 3A deletion in this area, whereas Illumina does not. And when we look at the Sanger results, we see that it supports Oxford Nanopore reference call here for A, same from the reverse strand. But then very interestingly, in case of this region where Nanopore calls for 3A deletion, in reality, it's just 2A deletion. So we call it that Sanger sequencing in this case mostly supports Oxford Nanopore case. But still, it's closer to Nanopore than to Illumina. And then one of the variants we looked at was a unique for Oxford Nanopore variant. In this case, Nanopore calls for alternative G here, whereas Illumina calls for reference T. And when we look at Sanger results, we see that in this case, Sanger supports Illumina call. So based on this very brief study, we can see that both platforms mostly agree. Still, each of them generates some unique results. Based on this particular study, out of 4 examples that we resequenced using Sanger, I would say that 2.75 were supporting Oxford Nanopore call and versus Illumina call. We think this is a pretty promising results, and we are going to extend this study to conduct a full-scale cross-validation using at least 10 clinical and reference samples to evaluate the full extent of concordance and discordance between these 2 platforms. In case we decide that it passes the validation, we will develop reporting standards and start to implement Oxford Nanopore testing in parallel with Illumina testing in our lab. So this is my story for today. And with gratitude to entire CIAN Diagnostics team, I want to extend my acknowledgements to Jesse Clark, who did all the bench work for both next generation sequencing and Sanger sequencing preparation, Kiran Adhikari, who is here for bioinformatics, and Dr. James McCardle for his constant support in reviews, discussions, and help with experimental planning. Thank you.
And who are your customers? Do you have the same NIH constraint?
No, we are a clinical lab, commercial lab, so our customers is anywhere. We have contracts with government as well as we serve just personal client. Anybody can become our client. Yeah, sure.
The study was pretty interesting. Do you have any reasons to believe why those were miscalled, and would they be consistently miscalled if you reread those samples with the same technologies again?
That's actually very good questions. I believe that the miscalls come from both sequencing errors as well as a bioinformatics constraints of a specific bioinformatic tools. For example, I can tell that in our lab with Kiran, we evaluated a number of bioinformatic tools for analysis of Oxford Nanopore data, and they weren't 100% agree between them. So depending on which tool you are using, you will always get some differences. This is why in clinical testing practice, if you detect any pathogenic, likely pathogenic variants via next generation sequencing, this is a must that you do a Sanger confirmatory testing as a golden standard for confirmation of those clinical variants.
Do you recommend that you use different analytical or informatic toolkits for each of your reads to find convergence across those calls?
I guess it will depend on your particular task, what you're trying to achieve. Typically, if you want to know exact sequence, you would do Sanger confirmatory testing rather than playing around with bioinformatic tools. So when you're validating a method, a test, that's what we do. We play around with different bioinformatics tools. We find the one that works best, and then we do the validation using just that particular tool.
These are all validated methods, or are they that are do you develop them in-house, or are they no validated?
So for clinical lab purposes, we need to do validation of any method, even if they were previously validated. We need to revalidate them internally to show that we can achieve the same accuracy and precision as it's supposed to be. But yeah, we do have, typically all these methods are called lab developed tests, but they go through a thorough validation process with validation report written to demonstrate the accuracy and precision.
Will multiple sequencing attempts with the same instrument produce similar variants, if you know?
Yes, I can show you an example. For any validation of next generation technology, that for those genetic tests that we developed in-house, for validation, you need to run each sample in triplicate and demonstrate that you get identical results. And this is typically the case.
With the same instrument?
With the same instrument, with the same technology, which you are validating. I think Bao was first.
No, just a quick question in terms of cost itself. Do you think that for the long run, the ONT gonna be cost less than the
Illumina? Yes. So based on our internal calculations, based on the full capacity. So if you use full capacity of Illumina sequencer, which is not always the case, but if you do, the cost for this particular test, the cost of nanopore sequencing about 3 times less, as well as a hands-on time is about twice shorter. But also that's taking in account full usage of Illumina capacity per run. As you go with less samples, your cost efficiency decline. So yeah, one of the advantages of nanopore sequencing that it's really, really cost effective.
So do you need at least two sequencers if the Illumina that initiates the example for validation for robustness?
For Illumina, so if we are validating Illumina-based test, we are validating it using one instrument, and that's the one that we use for production, for testing. I think you had a question.
You can take it offline.
wait.
Right. Yeah, you can say, after the presentation. Thank you.
So I figure we have about 5 minutes. That's what we need, but who's going to do a cesium chloride gradient today to isolate plasmids? Nobody. And the reason for that, how do I get this forward here? Next slide, push down. Is it going? Whoa, that's really slow. Very slow, is because most of you are probably familiar with QIAGEN. And QIAGEN actually has 2 sites here in Frederick County and another site down in the next county over in Montgomery Montgomery County? Down the street. So most people have been exposed to QIAGEN by either the blue box or red box. Blue box is for isolating DNA, red box for isolating RNA. What a lot of you probably don't know is there's actually a purple workflow, which is total nucleic acid. There's a green box for isolating protein. Or you've seen QIAGEN based on this. This is the spin column, which has turned 40 years old this year. It's a great way for isolating nucleic acids. But at 40 years old, when you're a company, you start to look around and think about what's next. And so what we've tried to do with next-gen sequencing is to make it easy and accessible to everybody. Just like taking a plasma prep that used to take days, making it do it in 30 minutes. If you look at a typical sequencing workflow, it's more than just the NGS run. So we saw the different sequencing machines and how you can build different libraries, but it actually starts with your sample. And I like the idea of no garbage in, no garbage out, or garbage in, garbage out. When we think about sequencing at QIAGEN, we actually start with our sample. And we want to preserve our sample. We want to make sure that we isolate nucleic acids or break open the cells in an unbiased way. Then we get to library preparation. Library preparation is the part where we add adapters and things to the DNA, RNA, or whatever we're looking at the sequence, and there's some special things we can do there. Then we do an NGS run, and then we have data analysis and interpretation. And what we've done at QIAGEN is we're a toolkit provider. We've gone through each of these different steps, developed out various different methods. We make them for different samples, whether you're starting with fragmented RNA, fragmented DNA, single cell, lots of things, an entire organism or single cells, and then we put them into workflows. When we think about a typical workflow, this is a workflow that we would present for total nucleic acid. Here you have solutions that are either automated or manual. You can go through and start with your sample, build your libraries, have some QC steps, run it on an NGS instrument. We're platform agnostic, Illumina, Element, anything else that could be out there. And then we have data analysis and interpretation. The way that we approach most of our customers is we either present a complete workflow or customers go through and they pick a la carte, what is necessary for the research. When we look at library preparation, this is the part where we take our nucleic acid and we're going to prepare it for sequencing. And there's a lot of different steps in here and places where you can introduce bias, place where you can go through and have something that maybe is going to cause a problem in your interpretation. So we've come up with various different methods that are enzymatic or different ways that we can fragment DNA and RNA. We have blocking ways that we can remove ribosomal RNA and other RNAs you may not want to sequence, such as yRNAs. This increases the sensitivity of assays. We have several different reverse transcriptases that we use based on the application. We have heat-stable reverse transcriptases that work at higher temperatures for certain things, or sometimes we have lower temperature ones. When we add the adapters and all that part that makes your library ready to sequence, we have many different adapter configurations that work with different instruments, different barcodes. We also have something which we call unique molecular indexes. These are little barcodes that we add to the individual molecules so that when you go through and do data analysis, we can do error correction. We can look at PCR artifacts. We can look at things that get introduced that are not part of the sample. When we think about library construction, there's a lot of different ways to do it. You can build a library for whole genome sequencing. You can focus on a very small little piece. You can look at different regions, and the number of targets you want to look at may vary. And that's why when we developed out our portfolio, we developed it out based on the amount of sample that you have, the quality of your sample, and then what you want to do with it. So for library preparation, we sequence everything. But we can also use a double primer or an amplicon, a single primer, or probe-based approaches to enrich for part of that. And that allows us to enrich for part of the genome that you really want to sequence so you can run more samples at one time. And that can be done on both a short read sequencer like Illumina and Element, or even long read sequencing. So you can enrich for different things you want to sequence there. These are some of the things that we're trying to control as we go through and we do our workflow. So we're trying to control about GC content. If you're looking at microbes that might be 20% GC to 80%, we want to make sure we get an unbiased representation of that. We want to make sure that we go through and we look at anything that can modify the nucleic acid starting at the beginning and make sure that we remove any sort of errors that we have in there. So we often have spike-ins and all these other things that work in the library preparation stages. As an example of what library preparation is, this is how you would go through in the lab and do a cooking exercise to sequence microRNA. So starting at the beginning, we're going to isolate total nucleic acid. And then we're going to go through a series of enzymatic steps, make cDNA. During some of these steps, we do what are called bead cleanups. And then at the end, we have a library that we can sequence. What you don't notice here is actually we've incorporated some different things in here like unique molecular indexing. So every time we make a cDNA molecule, it has a very unique barcode that allows us to go back and do some error correction. And we're also blocking yRNA. So as a research scientist, you may not realize what's going on here, but when you get your data, you'll have a higher content of microRNA reads because you're not sequencing yRNA, and you'll be able to know the number of individual molecules that were sequenced. And that really helps us go through and do the data analysis when you're starting with something that you can't even measure from a serum or plasma sample. This is our kind of workflow for looking at gene and non-coding RNA. It's various different ways that you can build libraries. So you can build a whole transcriptome by doing RNA removal or mRNA enrichment. You could do 3' sequencing. You can enrich with different methods. And so for a lot of the customers that we work with, they have some sort of specific question in mind. And we often look at the sample type, how many samples they want to run, what their budget might be, which is always very small, and how can we get the most information from their sample in the most cost-effective way. Here's another example for total nucleic acid. So I had talked a little bit, or I think the title of our talk was, how do you go through and do liquid biopsy and low biomass samples? And one way you can do a low biomass sample is not split it. If you want to get an RNA and a DNA signature from something, often you go through and you isolate RNA and DNA and you build 2 different libraries. You go through and then you sequence those in parallel. Here's a way that we can take one total nucleic acid, which is I call the purple workflow here, and we can build an RNA and a DNA library at the same time. We incorporate in these unique molecular indexes here, and then at the end we split it. And we split it because we want to sequence the DNA and the RNA library, maybe with different depths or with different configurations. But at the beginning, we've captured all the information at the same time. So this means if we have a small little piece of an FFP sample or a small amount of tissue to start with, we can go through and preserve all the information that's there. Here's another way that we can go through and address samples, and it's not so much the sequencing, but it's the method we can take fractionate a sample into its components. So here's an example of starting with 2 times 10 milliliters of blood, and we want to go through and do a thorough genetic analysis and a transcriptome analysis here. It's a kind of a workflow that we put together where we start with one of the 10 milliliter tubes. We can go through and we can look at circulating tumor cells. And we can go through and look at the circulating tumor cell total nucleic acid using a TNA approach. Then we can also go through and break this up into circulating free DNA. We can do genomic DNA. And with the second 10 milliliter tube, we can go through and do a complete transcriptome. So with each of these different methods, we've established a library preparation kit that will allow us to get the most information out of each of these fractions. So in the essence of time here, these are all the different workflows that we have at QIAGEN, which have multiple library preparations around it. It's a pretty large catalog. And usually when we think about going through and doing sequencing, it's not how we get there. It's the whole workflow that we're concerned with. So with that, I will adjourn this part and we can go have some coffee and no beer, I guess, today. Coffee and pastries. And thank you for your time. Yes.
For your liquid biopsy samples, you can grab the purple workflow.
Yeah.
Will the DNA and RNA are ready for long read technology as well, or it's just going to be applicable for short read?
We've made it for short read right now. Yeah.
So in terms of yield, does it affect the yield if you do it separately?
Total nucleic acid. Yields are very similar to doing an individual isolation of RNA or DNA. With this particular total nucleic acid, we also get a protein fraction. So we have, the nucleic acids and the protein, which can also be mass spec'd later on to add to the data set. But the yield of total nucleic acid is similar to doing an RNA and DNA. With the total nucleic acid workflow, you can actually do just an RNA or DNA workflow, or you can take RNA and DNA and put it together. So it's kind of a multi-use workflow. You can do whole genome, whole transcriptome, or you can do hybrid capture behind those workflows as well. Yeah, yes, question.
With that, do you notice that I'm assuming that all this is both for a multi-element sort of perspective.
Yep.
Solution finding at the end. Do you see any difference when you do have those combined?
Yeah, so when you combine analytes, it's really important that you look at the entire workflow and you maximize, or you have the right sequencing configurations and the right number of reads. And you have we've always added molecular barcoding to things that we do that, so that we can use the UMIs for error correction as well as PCR duplicate removal. If you go through and a lot of our data that we have, we can talk about this later, is we have the RNA workflow by itself and the total nucleic acid, and the DNA by itself and total nucleic acid. And we've optimized things so the data should be virtually the same on an Illumina or Element sequencing machine. Yeah, that's really important that we preserve the data. So even though we're combining things. Any other questions? No? Okay, thank you.
Okay, thank you so much. Our amazing speakers for today would like to, then our audience and folks online, and thank you for all your great questions. And I would also like to express our gratitude to our sponsors. The main one is Protein Tech, and then Advantage Tech, and McBee, Moore, and Vanik, our returning sponsors. So for the great breakfast from Panera, please feel free to enjoy the coffee and the pastries. And if you want to stay and chat with the speakers or with each other, you're welcome to do that while you're enjoying your coffee. So our next Biotech Connector event will be at the end of February next year. So more details to come. Thank you so much for coming.